So in this video, I'm continuing on with the series on multiple choice questions concerning RNA and DNA structure. So like I said, it's part 4. I've done 3 parts before that, answering the questions I'm about to show you. So that's part 3, this is part 2, and then part 1. So if you want the answers to those questions with some explanation, I highly recommend you go check them out because I explained them, so you won't have to memorize anything. You should understand it after that. But also, lastly, make sure to check out the videos I made on DNA and RNA, where I actually explain the differences, try and give you good ways to memorize things, and all that, because then these questions will be far easier. Okay, so let's get on to the question. This diagram shows part of a DNA molecule. So like I said with all my other questions, I really recommend you approach the situation by not looking at the question just yet. Look at whatever diagram they provide you and make sure you understand what's going on before you start putting answers into your head from this, okay? Because that might put too many things in your head at once. First, look at one thing at a time. So you approach this diagram. What I normally like to do is approach the diagram. That will make things far more easy. So what I can see in this diagram is they say it shows part of a DNA molecule, and we know that it's true because DNA molecule consists of 2 strands. So this is one strand. They only show 2 nucleotides. This is one nucleotide. This is one nucleotide. This is one nucleotide, this is one nucleotide. So they only show 2 nucleotides on top of each other. Obviously, DNA continues on up or down for a long time. It's very long, right? So we know it's DNA because there's 2 strands, 1 strand and 2 strand. Because RNA would only have 1 strand. Okay? So that's just confirming what the statement is. Now, each strand is made up of nucleotides. That's basically one subunit, because this is another one, right? And then they continue on. So one nucleotide, one subunit of DNA is made up of the circle, which is phosphate, the pentose shape, which is the sugar, and the rectangle shape, which is a base. Or this one will hold the letters, you know, A, T, C, G, those letters. The rectangle basically holds the letters of the DNA, the letters of the alphabet of DNA. Now these 3 shapes, you can see, are connected to each other by lines. We call these lines bonds. They are bonded together so they don't float around randomly. Okay? Now these bonds that hold together a nucleotide, or even different nucleotides together, so this is one nucleotide and the one below or above will also be held together by a line, a bond, right? Now this kind of bond that holds nucleotides together inside each other, so all the different subunits together and different nucleotides together, that kind of bond is different from the bond that holds nucleotides together side by side. So this kind of bond, that's what I mean. So this bond is different. The bond that holds nucleotides together side by side, so left to right, this one, is different from the one holding the subunits of nucleotides together and nucleotides above and below each other. Okay? So this is important to know. Now, that's it for looking at the diagram. We now looked at the diagram, we know-ish, and we can see they're pointing to X, so they're pointing to this bond that I was mentioning. That bond, this bond here is the same as, for example, here or here. It's the same kind of bond. So let's see what the question is asking. What type of bond does X represent? So like I said, this bond, X bond, is different from this bond holding left to right together. So let's look at the answers. A, could it be a covalent bond? B, could it be a hydrogen bond? C, a peptide bond? Or D, a semi-conservative bond? I'll tell you right now that it is a covalent bond. And let me show you how you can remember that. We know it's not a hydrogen bond just because, try and bear in mind for this because this will make things a lot easier for you. So a hydrogen bond is the one holding things together left and right. Normally, this is important, pay attention. So normally they represent hydrogen bonds by dotted lines, like I did in my previous question. See, they normally use dotted lines. Almost always, 99% of the time. But sometimes, and this is easy to remember, so it's easy to remember hydrogen bonds using dotted lines because I consider it's like it's a hiding bond. It's dotted lines, so it's a weaker bond. So hydrogen bonds holding the left and right together is hiding bond. It's not so strong, it's dotted. That's normally how it's easy to remember because 99% of diagrams will use dotted lines. But in the case where they don't, like this one, where it's a straight line, it's not dotted, it's hard to remember. So remember that the ones holding the two strands together, so the left strand and the right strand, is called a hydrogen bond. This is it's harder to remember if they don't use dotted lines, but I promise you 99% of the time they use dotted lines, so using this idea of hide is easier. All the rest of the bonds, all the bonds except the ones holding the left strand together and the right strand together is covalent bonds. So this one here, 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 all of them, legit all of them except the ones holding left and right together is covalent bond. Okay, so I can just point to the X. Remembering that the hiding bond is called the hydrogen bond is because it's actually weaker. So the bond holding the two strands together is weaker, and that's important because we want a weaker bond so that the two strands can be split open when you need to replicate your DNA. If it was too strong, we wouldn't be able to replicate our DNA, which constantly happens in our body. Okay, so that's why that one is weaker. Now the covalent bond is here, and it needs to be strong. It doesn't need to be broken at any point of our body, any point of DNA replication. It doesn't need to be broken. So just consider this would be a harder one because of the fact that the bonds seem the same. That this X bond seems the same to this one. So I'd consider this a harder question because they don't make it so easy to show that this is a different kind of bond. So the answer is covalent bond. Now next, I'm doing two here because they kind of relate to each other. Now let's go to the next question. So what links the pair of complementary bases in a DNA double helix? So we can just use the question from the previous question. So a double helix is basically the two strands, so this strand and this strand linking together, okay? And they twist, but that's not important for this question. They're asking, what links the pair of complementary bases in a DNA double helix? We know these are bases, these two rectangular shapes. The bases are basically like the letters of DNA, the alphabet letters of DNA, what makes you, you, the thing that codes for you. So they're asking what binds them together. We know it's a hydrogen bond. It's the weaker bond. It's the one that's kind of hiding in most diagrams. So it's not a covalent bond because I remember that one does not link bases together, does not link the two strands together. It links things within each other, the nucleotide subunits together, or the nucleotides above and below each other, not ever the two rectangles together. So in this case, the answer would be hydrogen bond. Oh yeah, and by the way, so for this one, I forgot to eliminate the wrong one. So remember, we know why it's not hydrogen bond, because that's in the middle. We know it's covalent bond. A peptide bond is not related in DNA. A peptide bond is related in proteins. And this is not a protein, it's a nucleotide, okay? That's why C is incorrect. D simply does not exist. Semi-conservative bond does not exist. This is just a made-up word to make you think that it means something, but it doesn't mean anything. Semi-conservative bond is not related at all, doesn't exist. Now, let's eliminate the wrong ones for this one as well. So covalent bond, we know it's not that one because it's, I showed you already. Hydrogen bonds, we know that's correct. Ionic bonds is not correct in this case. Ionic bonds are not relevant in DNA either. And again, they use a peptide bond idea. This one is relevant in proteins, not in DNA. So always remember, the things involved in DNA and RNA is these two, covalent and hydrogen bonds. Never, if they mention any other bonds, it's not relevant. So I hope this made some sense, and I hope I explained it a bit. And one key idea I want to mention right now is that in the IB, questions repeat and repeat and repeat. As you can see in the series I'm making, I've grabbed all these from IB just randomly, and they're all so similar. They all use these similar diagrams. So if you can go through the videos that I'm making, these questions, and just listen to the explanations, I guarantee you nothing can surprise you, because I'm going to bring up all these questions and you'll even notice yourself that they repeat and repeat. And once you get used to all the different variations that they make, any kind of new question in the IB that they make shouldn't be of surprise to you. So I'll continue and make some more parts to this so you can study them before your test.